So Dr. Degui Zhi, I apologize, I know it's probably not the best pronunciation, is a Glassell Family Professor of Biomedical Informatics and founding chair of Department of Bioinformatics and Systems Medicine at the McWilliams School of Biomedical Informatics at the University of Texas Health Science Center at Houston. Dr. Zhi is also the founding director of the Center for AI and Genome Informatics. He received his PhD in bioinformatics at UC San Diego. Before joining UTHealth, he was a tenured associate professor of statistical genetics at University of Alabama at Birmingham. Dr. Zhi is interested in developing AI deep learning and informatics methods for biomedical big data. His team developed multiple generalist deep learning frameworks for the modeling of biomedical data, including Med-BERT, a clinical foundation model for structured clinical data, Gene2Vec, a distributed representation embedding model for genes based on their co-expression patterns, and unsupervised deep learning models for driving endophenotypes for genetic discovery. His team also developed advanced PBWT-based data structures and algorithms for population genetics informatics. So without further ado, thank you very much, Dr. Zhi.
Thank you for the invitation and thanks for coming physically. Let me adjust my camera a little bit. So yeah, I'm very excited to visit here. DBMI is one of the best places to do biomedical informatics. So I'm just going to share some of my recent research. So what my research do is more than just what I covered today. So I just take the liberty to share the big overview. I'm got a PhD in bioinformatics and I enjoy doing interdisciplinary research. So I try to find where the multiple things will intersect. So that's why the Venn diagram shows. So I do population genetics informatics that is using efficient algorithm to handle increasingly large biobank genetic data. And I also build the EHR foundation models, the clinical foundation model to read the electronic health record data, structured data, and then build the predictive models from there. That may be akin to the interest of many of you in biomedical informatics. But the topic of today is really on the intersection of AI, imaging, and genetics. It will be on deep learning immunogenetics mostly. So that's where I'm coming from. I witnessed the explosion of genetic discovery, found really powered by the thing called the GWAS or genome-wide association studies, which basically have a cohort. You have phenotype and genotype, and you do massive regressions for each and every marker against the phenotype and find the p-value that is significant after adjusting genome-wide multiple testing. And based on this simple statistical approach, people has really find a lot of genetic information, and it's really, this is coming from GWAS catalog. Many of you may know. We've about each and every chromosome, every gene, we find some genetic signals that is telling this gene is associated with some diseases or some phenotypes of interest. So it looks amazing. Almost the human genetics is done, right? But what I hope to convey is that there may be a new way of doing that in the age of AI. So this is an anatomy of traditional GWAS. We have a bunch of people, and each one we got some phenotype. Is nothing but a bunch of numbers, and the genotype again, bunch of number but lying through the genome, and you do regression. You got a GWAS. So this is cover mostly traditional defined numbers. What I hope to say, now we in a digital world, human has been phenotyped much more intensively through imaging, waveforms, video, EHR, omics, all that. Those numbers are not in the traditional realm of phenotypes. And usually people just use some existing tools to derive some numbers from those and then call it endophenotype and then run GWAS. And that's another like a large untapped resource of doing genetic discovery. So what I hope to do is use AI to do this process. Why? Because AI has been amazing reading images, waveform, video, EHR, and omics. So rather than human to tackle those things that is complex and we don't necessarily have a best idea to do that, we can leverage the powerful AI. So the main outline for today's talk is we just review where we are in terms of GWAS, and how do we can define new phenotype and some new statistics, and then I also discuss about the engineering challenge of running GWAS. So what is GWAS phenotype? Single numbers are, but I'm interested in brain imaging. And brain image, as you know, is a 3D volume image. Really, the human are not able to read the 3D volume. We have to cut into 2D slices, so we see some pattern in there, right? And also another way is to understand the brain image is we construct the surface based on the imaging data, or we can use diffusion MRI to figure out the connectivities among different brain regions. So all of these are new data modalities that are not simple numbers. Then can we do GWAS on this? So and traditionally we use image processing, take an image and derive some numbers. For example, for brain, we can derive measures like a volume of the hippocampus or some other regions. And for retina image, for example, we can define some sort of tortuosity and so on. So, but mostly just existing software. And when we pair with genetic data, we can do genetic discovery, right? The limitation of this is the phenotype are predefined, really defined by human, limited by the software we have. And the amount of information currently by human eye can extract from images is limited. And as a result, these phenotypes are limited as well. So people use supervised deep learning, which is learning from human. Human give a label, and then AI to learn from that label. It's power a lot of this domain, but the phenotype defined is still carry the same bias as human does. So what we do is we introduce so-called unsupervised phenotype discovery. Instead of using human labeling, we use unsupervised deep learning to let AI to define what's the best descriptors. So how? I mean, we really borrow ideas from the fast-moving AI field. So in supervised learning, there's at least two main approaches, not limited to two. One is so-called autoencoder or predictive model. So basically, you have an input, you have an encoder, and make it into some bottleneck we call endophenotypes, and have a decoder. Those are neural networks, and to reconstruct the input. And you just train the AI such that the reconstructed is similar to the input, right? And you don't need any human label. All you need to just feed the images over and over. And this is also called an autoencoder. Another way is so-called contrastive learning, which is you have an image and you perturb the image. And both runs through the same image encoder, and you want to make sure whatever on the other side, the endophenotype is similar after the perturbation. So in that way, you learn some robust feature of the input. This is also called a contrastive learning. We start out with the autoencoder. It is simple. We run through the UK biobank images, which is about 40,000 of them. We take 6,000 as training the deep learning model. We feed this encoder model, use 3D convolutional neural networks as the encoder and decoder. And we save the bigger sample size for GWAS because as we know the power of genetic discovery rely on the size of sample. So this is done by amazing master student Kush Patel and then my a junior colleague Dr. Zixian Xie. So they build this model for both T1 and T2. And this is example how the reconstruction look like. You can see it more or less the overall shape, right? If you zoom into details, it's kind of blurred, the lack of those gyro structures and stuff. So it's definitely not perfect. It is our first attempt. We use the mean squared error reconstruction, so it's not be able to got all the details. But nonetheless, we move on and we looking at the embedding of the UK biobank brains. We see we use the UMAP, nonlinear mapping to do to visualize. So each dot is a individual brain, and they look like a big block. But if we color by the volume of the CSF, the middle region, so we can see it do show some nice gradient. If we also color by age, gender and stuff, it will also show some meaningful gradient. So at least learn some obvious features of the brain, but there's will be much more we don't know how to color. And we run through the genetics, GWAS. So this is we have 128 dimension from T1 images and 128 to T2, so total 256 GWAS, and we aggregate the phenotypes, the Manhattan plot. So what the Manhattan plot shows is the strength of the association of all the markers along the genome against the phenotype of interest. The most significant p-value, the higher the peak, right? So we do see many peak down there, including some new loci that is missed by the traditional image-derived phenotypes, GWAS. And one thing you may wonder, like, yeah, we find some genetic information, but what are those phenotypes exactly? Frankly, we don't know. We leverage interpretability, interpretable AI to help us. So we leverage the so-called perturbation-based decoder. We inject some noise into some endo dimension, and then the decoder is translating a bunch of number into some image, right? So it actually is image generator. So we just use the original endo and the perturbed endo to generate original reconstruction and the perturbed reconstruction. So we repeat the 500 times, we'll know like which region of the brain that is often perturbed by this dimension. That gives us the idea what each endo is doing with the brain. I mean, it's again just to way of visualizing, but neural network can do more than just what we see. Right? Just to summarize this line for work, what I showed is using this new endophenotypes discovered by unsupervised learning, we compare to existing software developed phenotypes. We require very little processing of the image and very fast to run. After training the model, really fast to run and generate the endophenotypes that is having higher heritability, meaning they carry more genetic signal. And it's to our surprise, those numbers are mostly unrelated to each other. It is not by design, but I think post hocly, it may be just because I mean, the representation space of the brain is large enough to hold more than 128 dimensions. And also fascinatingly, we can use this to generate a new image, right? So it will give us some leverage to reconstruct and help interpretation. And with that first success, we move on to other image modalities. So this is from the diffusion MRI called fractional anisotropy. It's mostly highlighting the white matter structure. We run the same architecture and we see reconstruction also makes sense. It's blurred, but more or less the overall shape. And we run GWAS, it's got a bunch of discoveries. So really, we are no way expert in brain imaging, but we are able to uncover not only existing loci, but also new loci. Okay, and here, let me introduce another way, right? So the contrastive learning way of doing this. In this case, we use the Retina images. We train the model using the IPAC datasets, which is famous in Kaggle. We train a model that can embed this Retina image into a bunch of number. And then we just move that model without any fine tuning and stuff to UK Biobank and let it to generate a bunch of endophenotypes and run genetic discovery. So we actually started this project first, but it's finished later. It's the only constraints we use is a sort of metric learning. So for same person's pair of eyes, you know, they come from same genetic program. So we hope it will embed into some vector in the high dimensional space that is close to each other. And if a different person, they may be farther, right? So we just leverage these constraints and we train the model. The model is the regular ResNet model. And what we find is indeed for IPACs, we train the matched pair of eyes that is have a higher cosine similarity to each other than the random pairs. And the same model moved to UK Biobank also show the same separation. So it's validated it's a decent model. And we run this phenotype against the UK Biobank genetic data. And we also find a lot of nocias showing the success of this contrastive learning. So basically, we see this the two ideas. One is the autoencoder reconstruction, one is the contrastive learning. Both give us a good results. Yao Chen Xie is a student from my collaborator Jiwang Shiwangji's lab. So he think of why don't we just combine these two? So literally we just train one neural network by both reconstruction and the contrastive learning. So I have two loss function, right? So just simply doing so, what do you have? What do you find out is this is called the genome informatics, because the reconstruction the contrastive loss is use the informatics loss. So what we see is this is our autoencoder. We find some 26 loss. So this is only on top 10 PC. So it's just for the sake of simplicity. We found 26 loci, and there's a number of contrastive learning approaches is less powerful than that. But by combining the predictive autoencoder and the contrastive learning, our GIM model find 40 is more than any one of those. So it shows that the autoencoder and the contrastive learning actually give some complementary information of the brain. And then also recently we are thinking about this. So this is all the endophenotype pairwise correlation for our autoencoder model. It is if you look at the pairwise correlation, more or less blank. But if you really squint your eye, you could see there's a little bit of structure. But I will skip that part. One is more or less not correlated. It's actually nice for statistical modeling. And my colleague Han Chen, who is a statistical geneticist, he's thinking maybe you can do a treat those 128 numbers as a vector. So we really want to do the regression, not just 128 single variate regression. We do one multivariate regression. And that's that way we can aggregate the information across different dimensions. And after all, really the representation is live in a native 128 dimension space rather than 128 1D space. So when you do that, we do see like this is a single variant GWAS p-value, and this is on same scale the multivariate GWAS p-value. So can tell it's much more powerful. And with that, we are funded by the Alzheimer's disease sequencing project. And we build this model, and many colleagues are interested in replicating our results. Because in GWAS, initially I showed this very saturating picture, people running out of phenotypes. So all of a sudden we uncover number of new phenotypes. So people are excited, want to run on their own cohort and can replicate. So what I tell them is that there's several ways to replicate. One, we can just run our summary statistics against any other phenotype. There's multiple program to compare some stats across different phenotypes. And another, they could download our endophenotype we already shared at the UTHealth Biobank. So you can download those and then run your own multi-omics or GWAS, whatever phenotype you are interested in. And of course, you can download our model weights, the deep learning checkpoints, and then run on your own imaging data that require you to have your own infrastructure. But when you do that, you can generate your own individual phenotype and then replicate our results. And of course, if you want to replicate the methodology, you can get our source code and train your own model, and then even fine tune it. Maybe you can get a better checkpoint and send back to us so we can validate each other. So we shared our data and both the results, the source code, the weights. So feel free to check out deependo.org. And with those, we are starting to imagine a possible next phase of GWAS. So traditional GWAS, we have start from phenotype. We run the statistical test. We have a lot of loci. We do the post GWAS loci check. Really, we found are new, or I mean, if new is really exciting, but otherwise it's still confirming, right? And do a lot of bioinformatics checks against the databases literature, the database of other omics. And then eventually for the number of genes, you have a good confidence. You want to check the literature, how well my result is consistent with everyone. These are usually done by this first author, grad students, or postdoc. So it is mostly manual work. But for the entire pipeline, I can imagine if we want to scale up, there are challenges. So phenotype discovery, we show we use first model, we can simple convolution autoencoder, we can already identify those. So in fact, what I show here is a untrained 3D autoencoder, meaning we just randomly initialize weights without any training. We throw at the brain imaging. Guess what? You can find some genes already. It just because the CNN by itself encode some inductive bias of the image, right? But of course, if we train after training, we find much more signals. But by no means we have the best architecture, or we train the model to the best loss function. So we are experimenting and different ways of deriving these endophenotypes. And for genetics, for a statistical test, when we have a set infrastructure to derive those phenotypes, then we have a lot of phenotypes and we need to run GWAS. And in fact, during our paper revision, we were tasked to modify a little bit architecture and run the whole thing again. So we use the fast GWAS, which is pretty fast GWAS software, but still it take about 100 seconds per phenotype. And it translates into 7 hours per 250 phenotypes, and we need to repeat it for number of different settings. So it take a few weeks to run through our GWAS results. So it's becoming a bottleneck, interestingly. So we think of how to speed it up. So we use the GPU. Unsurprisingly, GPU is now everywhere and so powerful at the linear algebra. So we dump it's there, and then eventually we see it's finished much faster, about hundreds of thousands times faster because there's so many cores if you leverage right. So now the bottleneck becoming the IO, but at least for the running the regression, we have a much faster way of iterating. And now really the bottleneck becoming automatic bioinformatics check. So we usually check against the FOMA, which is very convenient online databases or softwares. You upload your GWAS summary statistics, it will dump like a post GWAS annotation to you. But it is built for single phenotypes, not for multiple phenotypes, not for hierarchical phenotypes, not for imaging phenotypes, and then need a better phenotype ontology. And we also check the GWAS catalog, are we finding new things or not? But becoming a large informatics work because again, we first need to match the phenotypes better. We also the SNP has correlations with other SNPs, and then what is new, what's not is becoming a very messy situation. So here's what I imagine. GWAS in the age of AI, right? We'll need to have a much ways to define new new architectures for new data modalities. And even after that, there's a number of like informatics challenges. We need a faster linear algebra to get a statistical test. So we develop a torch GWAS with using GPU. We also have a JGWAS for the multivariate. Those are scaling up current GWAS statistical tests. And after we find those loci, we need to have a faster way of checking against the known results, need the automatic bioinformatics check. And once we find those gene hits, we also need to check with literature and maybe large language models with retrieval augmented generation can help us to automate these tasks. And then those are towards imagine a future where we have an automatic genetic discovery engine. I mean, of course, it's not possible just by myself. I hope everyone, if you're interested, you can join us. Here's like in terms of scientific idea. We use the endophenotypes. We can interpreting existing brain GWAS results. Supposedly, previous result find a gene or SNP associated with some phenotype, and we through our GWAS find the same SNP associated with some endos, and then we know the same endo map to some brain regions, and then we'll generate a hypothesis that maybe that region is relevant to the genetic manifestation to the etiology of the disease. Right? So, for example, this famous locus for Parkinson's disease, without knowing what is Parkinson's, what region is relevant to Parkinson's disease, we know through our endophenotypes, it's one of this endo is associated with this SNP, and also this endo map to mostly the thalamus region of the brain. So, I mean, without knowing literature, we hypothesis that thalamus, putamen, pallidum may be relevant to Parkinson's disease. But of course, if you are expert of Parkinson's disease, you know these are indeed the relevant region. But we sort of naively, through our engine can give such a hypothesis. And similar story, we can find for Alzheimer's disease. So just a quick summary now. We use deep learning AI, generate the phenotypes from complex data that go beyond the traditional single numbers. And one way to look at is the GWAS, or the statistical test, serve as a genetic verifier of the AI generation. So AI just generates some endophenotypes, learn some feature, but is this really heritable feature? Is that really have some biological factor behind it? So we run GWAS. If we get some gene hits, meaning that it hits on something that is genetic and probably biological. And there are a lot of potentials for methods and application development. So we are working on other imaging modalities and electronic health records, but we are completely open for collaborations if you are interested in doing any aspect of this. So with that, I would conclude by thank you. This large collaboration from many experts in wide range of areas. For the brain imaging, we mostly funded by NIA, and under this project, those are the co-investigators. Expertise, we really integrate expertise in deep learning methods, biomedical imaging informatics, statistical genetics, bioinformatics, molecular biology, epidemiology, and medicine. And similarly, for the retina, we are funded by NEI. We are incredibly fortunate to have this large collaboration to sort of put this thing together. And also thank my team. This is actually my team at the end of last year, and this year we have about double the size, but don't have a good picture to put on yet. Well, with that, thank you. Any questions?
You mentioned the time to run the GWAS inference, etc. But what about the time to train the contrastive for the autoencoders model?
Okay, so the question is about the time to train the deep learning model and the stuff. Yeah, so actually initially we don't have a very powerful GPU. It take some time, but with the funding we got a A100 server, and then training the whole thing usually take about a few hours. And then we that enable us to run a different setup, hyperparameter training, loss function regime and stuff. Yeah, so it's no longer a big bottleneck. That's for training, so that why the bottleneck becoming even downstream to us. Thank you.
Dr. Xie, I have a quick question. How do you combine the two laws? One is contrastive loss. How do you combine the two laws together? You minimize the first one and the next one, or you have a weighted, give the weights to the two laws?
Okay, I understand the question. So you mean that's right? Really, for deep learning, for your training, you have to define a number called a loss, and the number has to be a scalar number, not a vector. So in this case, we have two loss, we have two numbers. How to combine two numbers into one number? What we do is just add them up, and then there's a small choice in what's the relative weights of the two. We can do some experiment, but usually just half and half. It can give some good starts. I forgot exactly what's the final weight we use for the two loss, but so long you can add those number up, and then you do the gradient descent, it will be able to train the model to minimize both loss.
Thank you. So another question is, can you explain more what is imaging phenotypes? I only hear, clinical phenotypes, like if you define the phenotypes as like a region of interest, or how do you define the imaging phenotypes? And also, is this architecture working for other imaging except brain imaging, like CT scans, like for other disease?
Yeah, good question. So really, that's a little philosophical. So what is a imaging phenotype? So we call it endophenotypes. I think from like computational point of view, is you just have a function, function parameterized by neural network or whatever software. But what the function does is input the image and output a number. So long you have such thing, that's called a phenotype. And in our case, we have a neural network and give 128 numbers, which is just, you can think of a multivariate function. And that's a more general definition of a phenotype or endophenotype. And what's useful for genetic discovery is that you will you have a data available that is have both imaging and the genetics, and you need a large enough sample size. We tried this on like a few thousand individuals, it won't work. You have to tens of thousands, and luckily we have a UK biobank who have both the imaging and the genetic data. And as to say what image it work, so long you have this large number of images, and we try on brain imaging. We have structural MRI like T1, T2. We also have this connectome, DTI images. They all work. And for retina images, we start with fundus photo, like I showed, it works. We are working on OCT images. Hopefully get some different results. And then really, I can imagine any imaging data you can find, so long it pair with the genetic data, there's potential to be applied by our method. Thank you.
Have you tried a similar technique across different modalities of images all together? So patient might have like a volumetric scan of the brain, but then the different types of scans. And the phenotype might be related to all of them together versus just one.
Yeah, that's a good question. So this question is about if we have tried simultaneously feed multiple type of images. It's some sort of multi-modal AI. The answer is yes, it's possible. We just didn't try that yet, but it's quite possible. We can imagine if say for the same person you get two images. In here is perturbation, but you can think of two type of images are the perturbation of the same person, and you feed them in, and you can learn some sort of contrastive loss that you want to learn something that is common to both data modalities, and then pair with genetic, you can do the disc. I think it's possible. We just didn't try that yet. Good question. Okay.
Question on Zoom.
Okay.
George.
George, if you want to mute yourself, yeah.
Yeah, sure. So I wanted to ask a bit about the dimensionality reduction methods you're using, in particular UMAP. And my understanding was you're also comparing the 10 PCs from UMAP in the initial results. So I was wondering, I know I don't work with UMAP much, but I know it's considered by some outspoken researchers to be a controversial approach. So I was wondering if you have any opinions for or against those critiques, and if you find that using UMAP instead of something like t-SNE or PCA would drastically change the results, either in your research or in the field in general.
Okay, yeah, I know there's some healthy critique to UMAP in the field. So we use UMAP basically just a visualization tool. We want to show this how this brain imaging embeddings are related to each other. So we color by gradients and by other features and show nice gradients of the thing. We were hoping to have some naturally forming cluster, but we don't. We don't find those, unlike single cells data. So I think that may be reassuring. If there are, there's a hard time to explain that. And in general, I think UMAP has its value. It's much more a flexible way of interpreting high-dimensional data. I mean, before we have a PCA, which is fantastic, but it's only allow linear mapping. So it is mathematically simpler and well understood, but it would be less expressive. And UMAP give you the some freedom about nonlinear mapping and review the internal structure of your data. So I think it's useful, but you do need to be careful when you're interpreting UMAP. I know that there was a critique about, say, well, what is dimension one, what's dimension two, what I even mean in UMAP. I wouldn't venture to interpret that, but what's used to interpret is the distance or the clusters among the stars. So what UMAP try to preserve is the neighborhood distance of the individual samples. If you see some sample cluster together in UMAP, likely they will be also close to each other in the high dimensional space. But if you want to draw exact conclusion, you better check on the original dimension if are they still close to each other. And
Yes.
yeah, and for also just and finish up with your questions, like we did try PCA and t-SNE, and so in our data, they all give us somewhat different pictures, but more or less it's a big blob of thing. And we never saw some like substructures in the thing. So and the UMAP looks nice and the uniform density is sort of a blob, so we use that. We do have those other results in our supplementary in the paper.
Thank you for the talk.
Thank you. And there's another question online.
Yeah, how Chen?
Hi, can you hear me?
Yes.
Yeah, thank you for the great talk about imaging genetics. I have a quick question. So if I understand this correct, the overall goal is extracting features from imaging data and then train them as an input and then to train the phenotypes. So what I'm thinking is that if it's possible to use like something like the vision transformer or currently developed domain-specific foundation model to do such things and to extract features from images and then train as the input.
Great question. So that's something I didn't include in my slide. We are still training as we speak. So we did start experimenting vision transformer, which is now become a popular vision model. It shows in ImageNet and other vision tasks that's similar or outperforming the convolutional nets architecture. So we did try that, and it takes a while to train. It is you know it is slower to train and take usually take larger samples to make it working. So we just take out some intermediate checkpoints, and then we find it is indeed getting some genetic signal. It's many are similar to what the CNN find, and many there are also quite a fraction that is different. So we are actually trying to understand that results, and hopefully we can report that in our next paper.
Thank you.
Thank you. Just another question online.
Yeah, hi. I just wanted to ask, how do you interpret the error that you might get from AI-enabled phenotype discovery in terms of the results, like how you interpret the results? Like, would it affect your interpretability or your confidence in what you find? And what do you think we can do to maybe quantify that, or like determine like how confident we are with the results given potential errors that might happen with AI?
Yeah, I know we don't want to just simply trust everything AI generate, right? So especially for generative AI, large language model and stuff. In our case, our framework is not fully automatically AI. So we use AI to derive these endophenotypes, but we will run through the rigorous statistical machinery. We run statistical tests and only pick those that passing most conservative p-value cutoff, namely Bonferroni. And so then by that, we just trust the statistical rigor to give us like a well-controlled false positive in our discovery. But in general, AI, I know like for our interpretation, we just generate some images, but I don't know how to say. There may be error. In fact, the TMAP images we generated, they are not always well behaved. Sometimes it gives us a nice punctuated region in the middle of the brain. Sometimes it gives more fuzzy patterns, and we don't know that's how to exactly interpret. But I would say so far we are doing this AI human collaboration, human in the loop, so we make sure it won't just generate anything. But yeah, it's a good question. So moving forward, if we more and more rely on AI as a co-pilot for our scientific discovery, we'll always hopefully have a way to check the AI generation. Thank you.
There's one last question in the chat.
Okay. Okay, so this is from Chunhua. So what Chunhua asks is how much endophenotype data are currently available? How much informatics work does it take to integrate endophenotypes data with the phenotype? How much endophenotypes currently available? So we publish the 128 dimensional endophenotype derived from T1 and T2, but in our back burner we have a lot of more. We actually try increase the dimensionality from 128 to 256 and even all the way to 2048. I mean, really, we max out our GPU memory by doing that. But we do see like more or less 128 give us a decent power for representing the information in the images. But it's another theoretical question, fascinating, that we don't know the answer. That can we when can we know we extract all the good information from brain image? There are a lot of information. We only starting to scratch the surface. I expect there are many more. And how much information work does it take to integrate endophenotype with phenotypes? Well, this I think partly because rely on how do we organize the phenotype data. We will check say GWAS catalog, which give us some existing genetic association signals, but GWAS catalog has most of the flat structure is people dump their papers summary sets into this. The phenotypes are not always the same, not always using the same exact definition, not necessarily on same population. And they sometimes only dump the significant result, like passing genome-wide significant. Sometimes they dump everything. And sometimes they dump a multivariate association, which is a very different animal than the single variant single variate association. So it is a huge amount of work to be done, frankly, I think, to better organize the existing phenotype association information, and then we can better interpret what we discovered, how that's related to the existing phenotype association. This is about the genetic side, but of course, if you, in general, how the endophenotype correlates with the phenotype data without necessarily related to genetics, there's much wider net. I think it mostly really we need to figure out some ontology or some knowledge framework to organize the phenotype and endophenotypes.
All right, I have 3 questions. So first one is technical, and then there are 2 theoretical. So I'm thinking about, so image data, this evolution network is totally isolated. Like, multimodal, maybe some other architecture is better. What about time series? Have you looked into what kind of architecture is better for time series?
That's a great question. Time series is another dimension. So that means like instead of looking at static snapshot feature of the person, you have at a time dimension. Then we need some sort of alignment of individuals, across individuals. How we say we measure the same thing at the same time point. And that's maybe different. The same disease may onset on different people on different age, right? And there may be different disease status. Yeah, that's a fascinating question. I frankly, I don't have any results, but I think it's totally future looking.
Yeah, I'm just thinking like some conventional method like Fourier transform may be useful. There's a time when you use convert to frequency domain, but also there are things like the generative model for videos might also be useful in this case. Okay, so that's then we haven't tried anything. Right? So the 2 other questions about the genetic. So endophenotype, my understanding is one of the reason it's powerful is that when we look at the disease, we're looking at the underlying distribution is actually when we look at disease, we're looking at the individuals from tail ends of the distribution. So if you do endophenotype like this, you actually look at the complete distribution, and that can increase power. But actually, do you have example of that? Like you find something associated with the condition, and then you look at the corresponding brain region, and then you actually can find subclinical phenotypes of individuals carrying this. Of course, you need to have enough of this, but maybe a single step might not be enough. So
yeah,
examples of showing that this is exactly where is the power.
Yeah, I mean, also, frankly, so far what we're looking at are all normal individuals, normal brain, normal retina. So it all have this little nice Gaussian bump. Yeah, that's not a huge question. It's if we move into more disease relevant endophenotypes, we're going to see some like non-Gaussian distributions. How to that I don't know. I'm looking forward to to do more data and then.
So in other words, what you're saying is that everything you look at is subclinical in this sense.
Yeah, I mean, so far, right, we
You
just.
looked at the association with the Parkinson disease, right? That these people are antibodies, but they don't have Parkinson's. In that case.
Yeah, we don't know their Parkinson, but we take a look at the banks is supposedly mostly healthy. There may be individuals, including some individuals.
Okay, so that's good. So the other question is also, so this is related to multivariate association, and one of the reasons why multivariate association is powerful is that a lot of SNPs, a lot of genetic markers, they are associated with multiple traits. If you by looking at those multiple traits at the same time, you also increase power.
Yes, definitely. So there are parallel methodology work in other fields like multiple blood lipids or psychiatric genetics. I know people looking at some sort of multivariate association. But Han Chen told me like they are not of this scale. There may be a handful of phenotype. We are 128 phenotype, which is much bigger.
But my impression, you actually, so you are also, so you have the embedding, right?
Mm.
Do you take the PCs from the embedding to do the association, or you just take the embeddings directly?
We take the embedding by itself, but we can do the PC and so on. But supposedly, if we do multivariate, then PC or non-PC, they are same thing. Just rotate the multidimensional data however you want, right? It will give you same association.
Yeah, the interpretation will be different though. Whether so if you do TCs, you end up with okay, these are the ones that are overlapping, and then there's also no it's real PC. But in embedding, you do not guarantee those the dimension.
Yes. So I mean, we start with the individual dimension. So there is a little bit information is that I mean the neural network learns bunch of number, but those numbers supposedly individual are those are basically neuron activation values. Those are since they gravitate toward individual neurons, then there are some somewhat biased towards like this meaningful to have a neuron to represent some aspect of the image. So it's I think if you do the PC and rotating it, it will give a different set of bases to imprinting those multivariate. It may be highlighting some principal dimensions better, but it may be just talking about some random rotations of the space. We don't know.
Thank you.